Good afternoon. Welcome to the next webinar in our continuing series from MVF Bioscience. My name is Susan Hendricks. As a staff scientist, I've been working closely with a number of C. elegans researchers over the past 2 years during the development of Wormlab. I'm joined today by Jeff Sprenger.
Hi, I'm Jeff. I'm Vice President of R&D at MVF, and I've been working on the Wormlab project with the development group for about 2 years, and I'll be demonstrating the software for you today.
In today's webinar, we will demonstrate the capabilities of the Wormlab software. Before the walkthrough, we'll spend just a minute to become acquainted with our company's history to give you a sense of how we came to be creating this type of software. Medium throughput and behavioral analysis of C. elegans requires sophisticated automated methods to quantify the movements of the morphology and dynamic movement. We're going to show you that as well as the various metrics that are calculated in the process of tracking these worms. We'll show you the video capture tools that are a part of Wormlab. And we'll also discuss how Wormlab could be used to detect the movement of other species as well. If you have questions, please don't hesitate to post them via the question window during the webinar. We'll address them as we come across them. And after the webinar, if you still have questions, please email them to info@mvfbioscience.com, and we'll address them via email.
Some of you may not have heard of us, MVF Bioscience. We've been in business for 25 years. Delivering software and integrated microscopy solutions to the research community in life sciences. Some of you will know us as Micro Brightfield. We have systems installed in over 2,000 labs worldwide. Neurolucida is a leading product for neuron reconstruction. We have Stereo Investigator for counting cells and marking or measuring areas in tissue. And Biolucida is a whole slide imaging server serving very large microscopy images across the internet and within institutions.
As the first organism with a completely sequenced genome, C. elegans is a powerful research tool. This small multicellular organism shares genetic similarities to many vertebrates, including a 38% homology with the human genome. The developmental fate of each of the approximately 1,000 cells is known. Further, the identity and connectivity of all 302 neurons have been mapped. Thus, C. elegans, with their small size and short generation time, make them ideally suited for high-throughput analyses. Currently, most methods to assess C. elegans' phenotypic behavior are manual and laborious to perform, which precludes medium throughput studies. We wanted to increase the throughput of these detailed studies. Wormlab is a commercial product that allows investigators to perform automated quantitative analysis of locomotory and other complex behavior of freely moving C. elegans nematodes. These assays are used in many areas of research, including those focused on neurodegenerative diseases, aging, and toxicology. Additionally, locomotion is a common screening tool for genetic assays. Wormlab quantifies worms moving on agar plates with or without the presence of a lawn. It has been designed to keep track of worms even after interaction events so that the identity of worms throughout the entire experiment can be known, rather than returning information about the entire population of plated worms. This software uses algorithms that fit the worms, which provides many individual details about the worm, including length, width, wavelength, amplitude, and among many others. Wormlab is able to detect and track a number of species of nematodes and at different developmental stages as well.
One of the goals with Wormlab, something that we tried to do, was to deliver a product. This is a commercial product which would have a long-term product life cycle. So we delivered the product about a month and a half ago, 2 months ago in March, and we see a product life cycle that can run 10 or 20 years, similar to our other products. So the advantage of that is that by using Wormlab, you get continued support over a long period of time. So as people come into your lab and leave your lab, you have a consistent analytical method and a product that's been designed to be easy to learn and easy to set up so that you don't have to gather multiple components from different sources, but that you have something you can quickly install and run on a Mac or on a PC. I'm going to go over the user interface on this slide first before we jump into the demonstration, just so that you know what I'm doing as I move around the screen. This first element here is a drop-down for the workflow category. You'll see that right here in the red box. What we do is we support multiple workflows. This is the tracking worms workflow. We have another one for validating the tracking of worms as well. These are the individual workflow steps. We go from importing a video sequence all the way down to saving a project. I'll walk you through those today. This is the main view where you'll see the video displayed and also the tracking results with a graphical overlay. On the upper part of your screen, you see the tab marked 5worms.avi. You can load multiple videos, each one appearing in a different tab, and then track each one of these within a project. So you can collect multiple captures of video and include them all in one place. At the lower left, these are the typical VCR controls that most of you are familiar with to control the playback of the video so you can inspect the results. And over here in the lower left, you see the frame number and the time point as the video plays. The toolbar at the upper left is for detecting and selecting worms, so you can interactively edit. And then finally, we have a couple of navigation controls, zooming in or out, or using the hand to pan around, but you can also use your mouse to do that as well.
Okay, so let's dive right into the demonstration. We're going to switch over to Wormlab now and walk through the process of loading a sequence and detecting the worms.
You should see on your screen now the Wormlab sort of application up with nothing loaded. We're going to walk through these steps from top to bottom. So the first step is to import a video sequence. I'm loading this AVI file. Wormlab supports multiple video formats from both Mac and PC. There's over 200 different codecs supported within our application. So we'll open this up. You'll see here the video is displayed now at the center of your screen. You can use your mouse wheel to roll forward or roll back to zoom in and out, and then you can pan back and forth. The VCR controls, you can start playing the video, and you'll see here the worms are moving. So you can also navigate while the worms are moving and look at things up close. And this comes in this is very useful when you're trying to inspect the results of the tracking later on. The next step is to set the scaling for the video sequence. So let me open this up. And normally what you would do, you'd start your video taking a picture of a scale bar, millimeter scale bar, which we can provide for you. So here I'm going to click this measure button, and I'm going to measure off 1,000 microns or 1 millimeter, and I'll just do this approximately. You would do this against a scale bar. So when I click that, you'll see here it's counted 39 pixels and set your scaling factor. We have a frame rate here. It reads the frame rate from the video file. You can also adjust the frame rate. Next, we have the adjustment of the image. So what we want to do is we want to set up the basic image processing to separate the worms from the background. Well, this is a pretty clean image, so it's quite easy to do. We'll show you some examples later on with images with more clutter in them. So you adjust your threshold back and forth. If you adjust it too far, in other words, you're over-thresholding, that's too much. This is too little. And then finally, if you hit something around where you did, I've got this mark here at 109, where I can see the worms, properly outlined. This doesn't have to be precise. You just have to get it close. You'll see here on the left where my mouse is that we have what we call dark worms on a white background. If you're doing fluorescence, you'd pick light worms on a dark background. And then we have some other image processing options here. These are for uneven illumination or to reduce some of the noise in the background. I won't be going through that today, but it's something that you can try on your own videos. The next step is detection. So I'm now on step 4 in the workflow. And if you look here at the lower left, we have this box called detection parameters. There's all these numbers with a range of the area and the length of the worm and a width to length ratio. You don't need to set these manually. All you really need to do is click on the types of worms that you're interested in, and these will be set for you automatically. So let's do that. We'll click on this first worm here and this middle one, and you'll notice that these are changing over in the lower left. Detection parameters are changing each time. So I've selected 3 worms. We have some advanced parameters where you can change how precisely you want the worms model and so on. We won't be going through those today, but we'd be happy to cover those with you if you're interested at a later time. Regarding tracking, you can either switch to tracking by moving this tab here, or if you're on this step, you can click the tracking step. All of these windows you can resize, and you can also undock them and redock them. So if you have multiple monitors, you can have these controls on one monitor and the video on a second monitor. I'm just going to go through and track 50 frames for now in the interest of time. You could choose to just track only the worms that I've marked. If you don't select this option, it'll look and try to detect all the worms in the image.
One thing that feature might be helpful for is if you have multiple populations of worms in your plate, so you've got a couple of different ages, and perhaps you want to find out the locomotory behavior of all of your younger worms first. So you could run the sequence detecting the younger worms, and then run the sequence again with only selecting the larger adult worms, just to make the data export and comparisons straightforward and easy. So you can tailor the detection depending on your experimental paradigm.
Okay, we also have another option here for backtracking. You can use backtracking. I've turned it on now. If worms become entangled, backtracking will help to resolve those worms. They actually backtrack them from after they separate back through the entanglement. So let's start this up. I'll click the Start button, and now you'll see here at the lower left. Now your video feed might be a lot slower than what we're seeing here, depending on how fast your network connection is. But we're tracking at about 1.5 to 2 frames a second. If I zoom in while we're tracking, you should be able to see this now in fairly high magnification. You can see that what's happening is every frame we're refitting this worm model. So we refit the center line, we're measuring widths all the way from the head to the tail, and then we're calculating the centroid. And then we're collecting all this data in each video frame, and later on in the demonstration, I'll show you that data, how you can access it and then how you can export it.
We calculate the head and the tail on the basis of both morphological features and the direction in which the worm has traveled. So you can see there that worm 4 initially had the head indicated as the tail, but once the software finished tracking and did the backtracking, it was able to correctly identify the head based on the direction that the worm was traveling.
So that head detection is automatic. So even if it starts out in the wrong position on the initial detection, it'll automatically correct it once the video is complete.
So let's go back to the PowerPoint.
All right, so what you saw during this demonstration was we imported some videos. So if you have videos in your lab, you can import them into Wormlab. We have an option where you can actually capture the videos with Wormlab using our camera interface. That's the screen you're sort of seeing at the right here. We have a video capture window, and we have these camera settings where you can adjust the exposure, the gain, and the brightness. You can set the frame rate. We have this option as well where instead of capturing at a continuous rate, you could capture every 10th frame or every 100th frame for time-lapse captures over a long period of time. The cameras that we're showing you on the left, these are the cameras that we support today. They're point gray cameras. These are high-quality CCD, which is a charge-coupled display monochrome cameras. They work beautifully for capturing worms. The interfaces that are supported are FireWire, both A and B, USB 3.0, and sort of the new standard, which is Gig eVision. That's 1 gigabit Ethernet connection to the camera.
We have a couple questions about what sort of data we're able to quantify from the worm tracking software. So let's look at that in detail. Let's go back to Wormlab.
We track this thing for 50 frames. I'm going to load this from scratch. We have a project where we fully track this.
So all 359 frames rather than just the first 50.
Right. So I'll skip through on the bottom here, and you'll see that every one of these frames is now tracked. First, I want to show you some of the display options, and then we'll go into actually viewing the data. So this little icon up here on the upper left where I'm pointing is the display options. I'll open that up. Here we can get rid of that. So you can turn on each one of these components of the display. I'll just do them very quickly in order. First, you can turn off the entire display if you want. You can turn off the marker for the head. You can change its color. Orange might be a little difficult to see, so I'll change it to red. This is the center point of the worm. This is the tail of the worm. This is a blue triangle. That's probably also kind of hard to see. Let's change that color. This is the track number, and you'll see we've tracked 5 worms. This is the worm outline, so the outer boundary of the worm that we're detecting. You'll see later on that when we do an area metric, we're measuring the area inside this boundary. This is the worm center line. You can turn that on and off. The worm track is the total distance that the worm traveled during the tracking. So if you have a look at here over the right, worm track number 1, I'll put the video back on, and you'll see that the centroid, or the center point, is moving along the worm track. And finally, the track baseline averages the sinusoidal track, so you can look at more sort of a direct path of the worm travel. This is this yellow line. Now, one of the things that you can do with this is once you've tracked your worms and you have this graphic display, you can choose to export this graphic display by either taking a snapshot. That's up here on the menu. where are we there? We take a snapshot, or we can export the video. So when you export the video, this little window comes up, and you can choose what frame range you want to export and what file. And then what happens is it'll export whatever is on your screen. So it'll take the worms video, original video, with the graphic overlays. If you only want to export a single track, then what you can do is you can delete the other worm tracks and just leave the one track on and just export that single worm. So the export feature is ideal to include these worm videos after their track in your presentations, or to include as supplemental materials for your publications. So that's for the display. Let me just turn off the worm track. And now what we're going to do is we're going to go to viewing data. Somebody asked a question. I'll just jump in right here. Said I didn't get the difference between the worm track and the worm baseline. So here's the worm track, right? You see that's the path of the center point. If we average that path, if we average out that sinusoidal track, we end up with the baseline, which runs through the middle of the sinusoid, if you like. So you get sort of a more direct distance traveled along the baseline. Let me jump over to view data. This is the second last step in the workflow. So when I click view data, what I get is a table where the worm track IDs are along the top. We tracked 5 worms, and the video frame numbers are down the side. So you can see if I scroll through here, I go down to 359 video frames. We have 3 tabs here. We have the worm detail data, if you like, 1 per video frame. Then we have a summarization of each track, where we take each of the metrics and we average them over all of the video frames. And then we have frame detail. So frame detail is if you take this down at the bottom, you take the scroll bar and you select the different points in the video, these numbers will change. They'll show you the information for that video frame. So let's skip back up here to the detail. We're showing you the position of the worm in each video frame. These are the X and Y coordinates of the centroid, or the center point of the worm. You can also choose to show the position of the head or the tail. In this analysis, or these metrics displayed, are the length of the worm. So that's the length of the worm measured in each frame. And keep in mind that you can always go over to the track summary and look at things like the worm mean length over the entire track, the mean worm length. This is the width of the worm. So this is the average width over the length of the worm. And you have to recall that what we're doing here, let me just move this over a little bit, is we're measuring the width at each point along this center axis. And there are some advanced parameters where you can change the number of sampling points. I think for this run we have 59 sampling points, and then we're averaging those. The next is the area of the worm. I talked about that before. That's the area inside the outer contour. We have the speed of the worm. This is the instantaneous speed. Now this speed you can look at is really the distance change of the centroid from frame to frame. And this is highly affected by the fitting. So typically what you'll want to do is use a smooth or a moving average speed. So here's the moving average speed, again for the 5 worms across each video frame. And then you can average this across multiple frames. So you click this up or down, average over 3, 4, or 5 frames. This is the direction of the worm in each frame, and where we take a direction. This is in radians. So we have the direction of 0 is immediately to the right along the x-axis. Straight up would be pi over 2 radians. To the left would be pi radians. And then, of course, 3 pi over 2 downward. The following is this is the wavelength of the worm in each frame, and the wavelength is measured as twice the distance between the inflection points. So from here to there, double that distance is the wavelength of the worm. We have the smooth speed. With smooth speed, it's a little different than moving average. We use a statistical method to throw out the outliers. So if there's any problem with the video jitter, any rapid change or gross change in worm speed would sort of be outside of 3 standard deviations of the norm, and then we would throw those out. Or actually they're included, but they're weighted at a much lower significance. And then we have bending angle. So if you draw a line from the head of the worm to the centroid and another line from the centroid to the tail, we'll measure the angle between those 2 lines. So if the worm moves through different conformations, for instance, if it moves through an omega bend, the head and the tail tend to become, or the angle between the head centroid and central tail line becomes quite acute. It might reach something like 30 degrees, pi over 6 radians, and then we can detect omega bend using that information. We'll show you that a little bit later. And then finally, we have two more here. I'll just talk about these briefly, and we'll show you these in some subsequent slides. We detect the reversal behavior and also the omega bend. So we'll mark each worm with the word reversal if we're detecting that it's actually moving backward, which is defined as the distance or the direction opposite to where its head is pointing. So having the head position marked correctly is important, and we showed you that that's automatically corrected while we track anyway. I think that's it for the data view. I have one more thing to show you. It's pretty important, is how do you actually get this stuff out of here. So if I go to the position information, you'll see I have an export button at the bottom. If I click this and you have Microsoft Excel installed, we can export the information directly into Excel. I think that's happening right there. This is the Excel spreadsheet. It creates a tab for you for the worksheet for position. So each one of these metric tables that I showed you can be exported.
Not only that, but the data can also be refreshed. So on the worm data tab, you can see that there is the export button, but right next to it is also the refresh button. So if you make changes to the data file, so say you no longer want information about worm 2, so that worm is not of interest to your experimental paradigm, you can delete that data and the data will automatically recalculate. That would also be true if you wanted to swap the head and tail, for example. All the positional data would automatically be corrected. Speaking of the tail, Andrew asked a question about what the endpoint of the tail is actually being identified by the algorithm, and we're marking the end of the worm basically at the anus, not the tip of the tail. And so that's why it appears that there is still a small portion of the tail that is not being marked or fit.
Yeah, I think what you'll find is that when you're imaging this, that the tail can be very faint and it can kind of come in and out of visibility or contrast. So what would happen if you track all the way to the end of the tail, you'd have it constantly changing worm length based on how visible the tail was. So I talked a little bit about behavior detection. On your left is a video of a worm that is going through an omega bend. We're measuring that bending angle. The chart you see on the right is a graph of the bending angle, and as it drops below pi over 2 radians down here, we're detecting the omega configuration or the omega bend. Similarly, in reversal, you see the worm here is moving to the left, and then it starts to back up. So what we're doing is we're looking at the smooth speed, and when the smooth speed drops or goes into a negative direction for a certain period of time, then we mark the worm as reversing behavior.
So to summarize some of the metrics that we do calculate, we calculate metrics related to the position of the centroid, the head, and the anus, as marked as the tail, the velocity, direction, length, amplitude, the wavelength of the worm as it's moving. And we also detect a couple of behaviors, including omega bends and reversals. And so those are behaviors across a number of video frames in order to be classified as an omega bend or a reversal. This data can be exported directly to Microsoft Excel, and you can export the AVI file with and without video overlay so that you can put those in PowerPoint presentations or supplemental materials for publication.
The data that we're capturing, we also save in an XML format. So if you're interested in developing custom analyses, the position of the worm in each video frame is saved in XML, and you can import those into other pieces of software, things like MATLAB and so on, and create your own analyses. We talked a little bit about reversals and why reversals were important. Sue, do you want to talk a little bit about sort of the touch receptor response that some people are doing?
Sure, absolutely. So